I've always loved science. I always wanted to understand how things worked, how we came together. I was obsessed with space as a little kid. I was just overwhelmed by the enormity of it all and how little we knew. And I think as I've gotten older, I realized that yes, we know so little. My favorite type of science is neuroscience, and the more I research, the less I know, and I think that's quite humbling.
I remember watching a Discovery Channel program, and they were building these cars, and they were talking about the computers that they were using in the cars and how they were automatizing everything around in the car. So and then I got the idea I could do something like that, and not necessarily with cars, but with bioengineering or other kind of things. So that's how it all started.
I was trained as a medical doctor in Myanmar. I was wondering what if we can develop the new treatments which will make their life better and which can treat their diseases better than the current treatments. So that's what interests me into doing the medical research at first place.
It came about from, I guess, my love of animals. I just love animals and the diversity of animals, and that's how I initially got interested in biology. And I guess, you know, when I was in high school, I took some biology classes, and yeah, that's where I really kind of fell in love with biology.
I look more at science as an adventure into the unknown. And you know, you want to share the journey with people who can have a laugh and can enjoy the ride.
I'm Sally Mccarthy. I'm a Professor of Biomedical Engineering at Swinburne University of Technology and a Science Leader at CSIRO. My job is to bring everybody together, get the excitement going, come up with the ideas and help build the connections. And the really exciting opportunity with this program and the people I've brought together is building something new and exciting in collaboration. So I specifically selected people to join the team because they had an outward facing point of view on everything, but also they had very specific skill sets. So in the case of Sorel, who's come to us via Argentina, Colombia and Brussels to end up here in Australia, she's an electrical engineer. She loves building stuff.
I will grow the cells on this surface. They are made of gold. That's what I was working on yesterday.
Her idea of heaven is the fact that we're getting transistors and she's going to get out and build a system for her. Then we've got Zayar, who's from Myanmar. He's a medic in his background, but he's also done a lot of research and he's just finished a PhD. So he's fresh out of a PhD. He's from an Asian country. He's very quietly spoken, very respectful, but has the brain the size of a planet. So we're all learning we have to stop and listen really closely because what he says is always spot on. So Aletta, she's a biologist. She comes from a very traditional biology background. She's quiet, but very strong. So knows her stuff and is really willing to get out there and push some boundaries when she is feeling confident.
And there's so many papers out there that have done it in different ways.
Yeah.
Like if
Daniel, outwardly incredibly confident, physicist, chemist, knows how to build stuff, incredibly inquiring and talks through all the problems. So he's going to keep talking until he's come through to that way of thinking. He's driven by curiosity. And so one of the exciting things with working with Dan is that he's coming from a non-biology background to work at the interface with biology. And so he brings the questions that maybe we gloss over, and so we can dig into the problems in a bit more depth.
Resveratrol's metabolized. It goes in, cells metabolize it.
And then there's Charlie. And Charlie, she's got a really good background in neurobiology, but she's also got a really strong interest in engineering and maths. And so that's a really unusual combination, and that brings a really interesting flavour into the research we can do.
Because this layer is the rubber seal, right?
She's got a really great ability to analyse what she's reading, and that's really exciting, I think. For the whole team, she becomes a bit of a font of knowledge. Did you read that paper? How did that go? And so for all of us, it's each about engaging others on their way of engaging. It's not about everybody going into their little silo and staying in it. It's about a team forming that can bring their questions and their ideas and is confident in asking. There's no dumb questions. So at the moment, when we're working in the lab and we're trying to understand how our body reacts to things, we grow some cells that may have come out of the body at some time. We grow them on a flat piece of plastic. And then we can do chemical tests with them. We can explore whether the materials that we put in our body, say a contact lens, might have something come out of them and causes those cells to be damaged. So that's called two-dimensional cell culture because the cells are growing on a flat surface.
And this model is very useful and very convenient, and so a lot of people use it. But when you actually compare this one layer of cells to the human body, where you have like whole lots of cells growing in multiple layers and interacting with each other, so this is very different system from the human body. So that's why we're trying to bridge the gap between these two models by building a three-dimensional cell culture system.
So now the cells aren't just growing attached to one surface, they're growing in a scaffold. And that means that you can create tissue replicas.
So then once we try and do like treatments on that, we'll get more useful information. And that'll mean that further down the track, less clinical trials will fail because they're based on fundamentally accurate models.
And so the whole system's living, but it's not the whole animal. So we've sort of got this intermediary stage, and that's we're calling four-dimensional cell culture because the cells are grown in 3 dimensions, but we're able to look at them in time.
Right now, they grow these cells blindfolded. So they don't know what's happening. They cannot see the cells because it's not a monolayer anymore, but it's a tissue. So if they put it under the microscope, they cannot see through the skin. My idea, or Sally's idea, is to create sensors in this tissue. So when they are growing the cells, they know what's happening. If there is something wrong, they can stop and start again. So that's what I want to do. I want to make these sensors that are connected to the cells and that will give feedback information.
In the long term, what we're hoping to be able to do is to enable personalised medicine. So this would be the case where if we have a patient who has a tumour, we take a sample of the tumour tissue and a sample of the patient's healthy tissue, and then we can grow each of those tissues up into larger amounts and test them against a broad suite of chemotherapies, which will enable us to actually identify which drug is the best for that individual case.
And you'll also be, I guess, reducing animal use in science. We really need animals at the moment to discover new things, and so that's totally understandable. But yeah, 100%, if we could build models to reduce animal testing, I'm all for that, yeah.
I get my energy from people. From people having great ideas, from having fantastic conversations, so having really interesting conversations. I work with a great team. That's what gives me energy and optimism, is that, you know, we'll throw challenges up there, but we'll find a way.
Sally is really nice. She really gives you the freedom to work how you want to work. And she also guides you through it.
It's actually about helping people to find something that they absolutely love. And that, there's nothing better. There's absolutely nothing better than finding something that you're like, oh, this is what I want to do.
I do it because I like it. I do it because I want to build new stuff. I want to create new things.
I was in a hospital in cancer lab treating cancer patients. So we are still giving those treatments as a chemotherapeutic drugs, which were developed maybe 20, 30 years ago. There's not really any other options during that time. So we have to keep using these drugs, knowing that these patients will come back one day with a resistant problem. What if we can develop new drugs which will be effective in these patients one day, and that will save them millions of lives and makes millions of patient life much better.
I have a central nervous system disorder myself, so I have autism, and I was taken to a lot of child psychologists and neurologists, and I think I kind of wanted to understand why I was a bit different and why I thought a bit differently. And when I was little, I figured if I could grow up and study these things, maybe I could help people like myself or help people understand how I think and how other people like me think.
I just love research. I like learning new things. I like the feeling that you get when you discover something new. I feel that multidisciplinary science is the future of good science, and I want to be involved in that. I really want to have that experience.
If the work that we're doing is successful, it really could change the way that we understand human biology. There's a lot of hard work to do, and you spend more time with the people at your work than you do with your family, so in the end it's great to have a team that's worth spending time with. I don't believe that growth can continue forever, but the rate of scientific discovery is still accelerating, so that's pretty exciting. We're trying to explore big questions and big challenges.
Everybody has those challenges. You always have to prove yourself everywhere you go.
I look forward to us having our first device with skin in it and our first tests of infection. That's in my next 6 months. I don't know where we're going to end up, but I know we're going to have huge fun along the way. And I also think we'll ask great questions and we'll meet great people who will help us develop that vision.